Morning. Becky, you have a new computer? I can't hear you. First, are there any questions or concerns?
Okay, I do have one. It's just I'm borrowing one, so
I understand.
it's totally different than my other one. So hopefully I get that one back.
Okay. Are there any questions or concerns? Okay. So we should finish chapter 3 today and start chapter 4. You will see if you come down to this module exams, your first exam is posted. It will open at 1 p.m. today, or no, 12 p.m. today. It's open for almost 2 weeks. So it will close. How did I set it up? It'll close a week from yesterday, 2 weeks from yesterday at midnight. Is that, I have to remember what day is the 21st. That isn't 2 weeks.
It's
Yeah, it is.
Yeah, it
It's
is.
2 weeks from, okay, I'm like trying to do the math in my head and it's not working. All right, so 2 weeks from yesterday, and so it's going to cover chapters 1 through 3. So we will finish chapter 3 today. We will start chapter 4. Bear with me just for a second here. 57. So cell adhesion molecules. I don't want you to memorize each type here. This is just a figure showing possible cell adhesion molecule structures. But I want you to remember their functions. So they can work as molecular Velcro, anchoring cells to molecules in the extracellular space into other molecules. If you think, if you've talked about it in lecture, desmosomes, think we talked about it maybe in lab 2. Desmosomes. No, I think we talked about them last Friday, maybe. Desmosomes are a type of cell adhesion molecule because they work like molecular Velcro, anchoring cells to molecules. They also CAMs or cell adhesion molecules also work like arms. They're used in allowing migrating cells to haul themselves over past other cells. A lot of times cells will move, they call it diapetus. They will move past other cells. And this occurs via the use of these CAMs. Cell adhesion molecules can also be SOS signals on blood vessel linings that are signaling to white blood cells to nearby infected and injured areas. They can also be mechanical sensors that respond to tension at the cell surface by stimulating or the synthesis or degradation of different adhesive junctions. And they can also be intracellular signaling transmitters that are going to direct cell migration, proliferation, and specialization. One thing that's going to come up in the next slide is cell adhesion molecules are involved in what we call contact inhibition. What that means is that cells will actively divide until they come in contact with another cell. And when they come in contact with another cell via the cell adhesion molecules, they stop dividing because their division and their proliferation is based on the current cell population in that area, or the current cell concentration in that area, or the cell density in that area. Because cells will have contact inhibition to prevent overgrowth of cells. Tumor cells stop showing that contact inhibition. Any questions about cell adhesion molecules? So just like they sound, they're adhesion surface proteins that can adhere to other things. So cell adhesion molecules work like membrane receptors, and they're involved in cell recognition for normal development and immunity. They also can be involved in cell signaling by which they bind specific ligands, such as which we term first messengers. They bind these ligands, which can be neurotransmitters, hormones, paracrines, meaning things that are released by neighboring cells. And when the ligand binds to these receptors, it activates a signaling cascade within the cell. So the receptor binds the ligand. Remember that these are cell adhesion molecules. So this receptor that's a cell adhesion molecule binds to a G protein. And this G protein is activated, and as a response, it activates other things. one of the first things it activates is an effector enzyme. The effector enzyme then will go on to activate second messengers. So the first messenger was the ligand. G protein activates an effector enzyme, which then activates second messengers, one of which is possibly cyclic AMP. If you remember, we talked about how this was an adenosine derivative in chapter 2, very end of chapter 2. So this is one of the second messengers involved in a signaling cascade that can then activate multiple different possible cellular responses, genetic changes, metabolic changes, structural changes, and so forth. Any questions about cell adhesion molecules so far? One other thing is cell adhesion molecules can work like they can be involved in membrane channels that will function in result to a change in voltage. You remember we talked about that separation in charge across the membrane. So this is these channels open in response to a change in the membrane potential. Remember that one way to think about that membrane potential, remember the membrane potential is that separation in charge. And so why is it called a potential? I don't honestly know, but my guess is that it's because it's a potential energy source. It's a potential energy form where that's this separation in charge, where it's more positive up here, more negative in here, is a potential energy where if we let the current flow down the membrane, we can use that flow of current to drive other processes. These membrane sensitive channels are common in the functioning of muscle and neural cells. So from about, not this next chapter is on bone, but the following chapter till the end of the semester, we will spend a lot of time talking about muscle and neural cells. And these are key when it comes to the function of muscle and neural cells. Moving on to some easier things to understand and comprehend. From here on out, I think this chapter there's a lot of material, there's a lot of definitions, but the majority of it is, this is the cell structure and this is what it does, kind of thing. So a cytoplasm is the cellular material that is in between the plasma membrane and the nucleus. Remember, cyto means cell, plasm is anything else. Cytosol is a viscous gel-like transparent fluid in which other cytoplasmic elements are suspended. Organelles are the metabolic machinery of cells that are found in the cytoplasm. So the cytoplasm consists of the cytosol and the organelles. Inclusions are chemical substances that may also be present depending on the cell type. For example, they can be glycogen, stored nutrients, lipid droplets, pigments, water-containing vacuoles, etc. The mitochondria, the first organelle we're going to talk about, are the power plants that provide most of the cell's ATP through aerobic cellular respiration. Aerobic meaning it requires oxygen. Let me just for a second here. Aerobic meaning requiring oxygen. Oxygen gas or oxygen. Anaerobic meaning in the absence of oxygen. Or let's put it this way, when oxygen is absent. So mitochondria carry out cellular respiration. And through this cellular respiration, they generate most of the cell's ATP. There's two membranes within this organelle. There's an outer membrane and there's an inner membrane. The inner membrane folds within the interior, forming what are called cristae. With these two membranes together, we have two different compartments within this organelle. We have the inner membrane space, which is in between the two membranes, and we have the matrix that's in the inner membrane compartment. One thing that's very unique to mitochondria is they have their own DNA and their own RNA. No other organelle has this. This gives some very interesting, because of this, there's some very interesting ideas and a very interesting theory behind the evolution of the mitochondria. But it's beyond the scope of this class, so I'll just tell you it's out there. It has to do with one cell taking in this cell, and they join into a symbiotic relationship. However, the other abilities that come because of mitochondria having its own DNA and RNA is that if a cell's need for energy increases, the mitochondria can increase their number of cristae or increase the number of mitochondria within the cell, because they can actively do their own protein synthesis. So they can replicate themselves or add to the material within each organelle. The density of the mitochondria within a given cell reflects that cell's energy requirement. Muscle cells have an exceptional amount of mitochondria in comparison to other cells because their need for ATP is extravagant. Another organelle are what we call ribosomes. Ribosomes are made from two subunits that are composed of proteins and ribosomal RNA. These are the sites where protein synthesis is made. These are the sites of protein synthesis where they read the mRNA. mRNA meaning messenger RNA. So when DNA is expressed into a protein, first the DNA is converted into an mRNA sequence in the nucleus. And then the mRNA sequence leaves the nucleus and enters the cytoplasm. When it enters the cytoplasm, it becomes attached to the one subunit of the ribosome. And then the second subunit will come in and bind to the mRNA. And then protein synthesis will begin. Some ribosomes perform this protein synthesis while being free in the cytoplasm. Other ribosomes will bind to the rough endoplasmic reticulum and continue the protein synthesis. If the protein is made to be secreted outside of the cell or become part of the plasma membrane, the protein will be synthesized on the surface of the rough endoplasmic reticulum. If the protein being synthesized is going to stay free within the cytosol or in the cytoplasm of the cell and function within the cytoplasm of the cell and isn't made to be exported via exocytosis or become part of the plasma membrane, it will be made by ribosomes that are free within the cytoplasm. The endoplasmic reticulum is a network within the cytoplasm. There's an extensive system of interconnected tubes. If you look here, here's the nuclear envelope, which encloses the nucleus. Continuous with it is the membranes of the endoplasmic reticulum. And which includes both a rough endoplasmic reticulum with the associated ribosomes and the smooth endoplasmic reticulum that doesn't have ribosomes on it. The nuclear membrane with the membranes of the rough and smooth endoplasmic reticulum accounts for about 50% of the cell's membranes. So I skipped over it and then I came back to it, talking about proteins proteins being synthesized by ribosomes, either in the rough endoplasmic reticulum or free in the cytoplasm. Ribosomes make both of these proteins, like I said before. They make proteins that are going to function in the cytoplasm. They make proteins that are going to be excreted from the cell and enter the plasma, be excreted from the cell or be part of the plasma membrane of the cell. If a protein is going to be excreted from the cell or be part of the plasma membrane, as the mRNA is being read into an amino acid sequence, the protein, it will have a tag put on it, a signal sequence or a signal recognition particle put on it. If the signal recognition particle is on the protein that's being synthesized, that means that ultimately that protein is going to end up being part of the plasma membrane or be released from the cell via exocytosis. So if the protein is going to leave the cell or be part of the membrane, the signal recognition particle gets added. It binds to a receptor in the endoplasmic reticulum membrane. And from there on out, the protein is synthesized on the surface of the rough endoplasmic reticulum. Once its synthesis is complete, once all the amino acids have been added, that protein is going to enter the lumen of the rough endoplasmic reticulum. And then undergo a series of processes, which we'll talk about here in a second. Are there any questions about this? The other function of the rough endoplasmic reticulum is that it is the place where integral proteins, remember these are the proteins that can span the plasma membrane, and phospholipids are synthesized. So if you want to think about it, anything that's going to end up being part of the plasma membrane or be secreted from the cell is going to be ultimately be synthesized in the rough on or in the rough endoplasmic reticulum. So anything that's going to be leave the cell or become part of the plasma membrane is going to be synthesized at the rough endoplasmic reticulum. The smooth endoplasmic reticulum functions in metabolizing lipids, synthesizing cholesterol. And if you remember, cholesterol is the precursor molecule for all steroids. So the smooth ER synthesizes cholesterol, synthesizes lipid components of lipoproteins. Remember, this is a lipid protein complex. And all steroid-based hormones. So all hormones that are going to be created from cholesterol. It also absorbs, synthesizes, and transports fats in intestinal cells. In certain types of cells, the smooth ER has other functions, or there's specialized ER with other functions. For example, in the liver and the kidneys, the smooth ER also can detoxify drugs certain pesticides and carcinogens in the liver and kidneys. Can also break down stored glycogen molecules to form free glucose in the liver that can then leave the liver. Remember, glycogen is the stored glucose polymer. So it breaks down glycogen to allow for single glucose molecules to leave the liver. Another function of some specialized ER, smooth ER, is a sarcoplasmic reticulum, which functions in calcium storage during muscle contraction. We will learn, and we'll talk a lot about it, that calcium is very important for muscle contraction. Therefore, muscle has stores or a storage place specifically for calcium. This storage place is the sarcoplasmic reticulum. In addition to the endoplasmic reticulum, we have the Golgi complex or Golgi apparatus, which is a stacked or flattened membranous sacs that are associated with swarms of tiny membranous vesicles. This is the principal traffic director for cellular proteins. So this is this could be your traffic director or your postal service for the cell. It synthesizes carbohydrates and puts finishing touches on the proteins in glycoprotein synthesis. One thing that we should say here is that this receives all new proteins from the rough ER. Remember what proteins are synthesized in the rough ER? Proteins that are going to become part of the plasma membrane or will be exported from the cell. So what that means is that all proteins that are going to be, just to repeat this and make it really clear, all proteins that are going to become part of the plasma membrane or be released from the cell via exocytosis are going to be in the rough ER first and then be sent to the Golgi apparatus. In the Golgi apparatus, carbohydrates are synthesized, and some carbohydrates are added to proteins. So the Golgi apparatus is going to sort, cut, and splice proteins. It can add carbohydrates to proteins, and mature cisterns, little bubbles on the edges, can break off into vesicles and leave. Once the protein that enters the Golgi apparatus has had the finishing touches put on it, has been modified however it should be, it can then leave in the cisterns of the Golgi apparatus via vesicles. Once it leaves the Golgi apparatus, it can go one of three ways. It can either go to the plasma membrane and become part of the plasma membrane, or be secreted out of the cell. It can return to the endoplasmic reticulum, or if there's something wrong with that protein, it can be, the vesicle confused with the lysosome and be destroyed. So from the Golgi complex, it takes 3 different pathways. Either goes to the plasma membrane to become part of the plasma membrane or be secreted from the cell. It can return to the endoplasmic reticulum, or it can be that the vesicle confused with the lysosome and be recycled or destroyed. Any questions? Guys are kind of quiet this morning. The lysosome is what we call the stomach of the cell. What occurs in your stomach? Can you tell me a little bit about what you know about the stomach?
It breaks down food.
Breaks down food. How does it do it? What's
The-
pH like in the stomach?
It's very high. It uses the acids.
Let's say it's really low. So because you're right, Becky. It's the acidity is high, so the pH is low.
Right.
Okay. The lysosome is the same way.
Wouldn't it break? So it would break down the enzymes as well, right?
Yep. It breaks down. Well, it breaks down proteins, and a protein can be an enzyme.
Okay.
So this is a safe place in the cell where digestion can proceed. It contains digestive enzymes, which are called acid hydrolases. You know what hydrolysis means? To breaks bigger things into smaller things. So these are acid hydrolases, meaning that their optimum pH is a lower pH. And they can digest proteins, nucleic acids, carbohydrates, phospholipids, and other substrates. The lysosomal membranes have specific ATPase. Remember, that means that a pump that hydrolyzes ATP to pump something across the membrane. So it contains ATPase hydrogen ion pumps that gather hydrogen ions from the surrounding cytosol in the cell and pump it in to the organelle to keep the pH within the organelle lower than it is outside of the organelle. So in the lysosome, the pH is about pH of 5. Cytosol outside is a pH of 7.2. The enzymes, the digestive enzymes within the lysosome are inactivated at lower or at increased pH like 7.2. But at an optimum pH of 5.0, they break things down. And according to their name, you can look at the name and determine what an enzyme does. A nuclease breaks down nucleic acids. A protease breaks down proteins. A glycosylase breaks down carbohydrates. Lipases break down lipids. Phosphatases break down phosphorylated proteins. Sulfatases break down things with sulfhydryl groups attached to them. Phospholipases break down phospholipids. So these lysosomes work to retain dangerous hydrolases. They are good at breaking down things. We want them to be in a specific area, not be, we need them to be isolated and contained. So a lysosome keeps them contained while also releasing the recycled digested products. The lysosome is the cell's demolition crew. So it works to digest particles taken in by the phagocytes. Remember, those are the cells that engulf things. Phagocytes, being the cells that engulf things, tend to contain a lot of lysosomes because that's their function, is to take things in and break them down. Also are involved in autophagy, which means meaning to eat oneself. So their lysosomes are also involved in degrading and recycling worn out and non-functional organelles. They also perform metabolic functions like glycogen breakdown and glucose release. They also, when necessary, will break down non-useful cells. So when a cell is deprived of oxygen or excessive amounts of vitamin A are present, the lysosomal membrane becomes unstable and the lysosome ruptures, resulting in autolysis of the cell because those acid hydrolases that break down things are now not contained within the cell and are able to function throughout the cell, allowing for autolysis of the cell. Especially during development, we have systematic programmed cell death. And this is done through the lysosomes. For example, in human development, we have webbing in early embryos. We have webbing between the fingers and the toes that is systematically broken down to allow us for better dexterity of our fingers and toes. Also, the uterine lining is broken down this way through menstruation, during menstruation. Moreover, as we're going to talk about in the next chapter, chapter 7, bone is broken down in osteoclasts to release the calcium via lysosomes. There's a disease out there called Tay-Sachs disease. In Tay-Sachs disease, which is a genetic disorder, lysosomes lack a glycolipase that's necessary to break down glycolipids in the nerve cell membranes. As a result, the lysosomes in the nerve cells swell with accumulated undigested lipids. And whenever there's a swelling of an organelle, that organelle becomes bigger and bigger and starts putting pressure on other things within the cell and causing malfunction of other things within the cell. Remember how structure and function are very closely related. So if there's a swelling of a structure, it can possibly impinge on the function of other structures, resulting in a loss of function to other structures, which is what occurs here. In Tay-Sachs disease, this accumulation of glycolipids will eventually result in mental retardation, seizures, blindness, and ultimately death within 18 months. Peroxisomes are membranous sacs similar to lysosomes in that they contain a variety of enzymes that are important in detoxifying harmful substances and neutralizing free radicals. You remember in chapter 2, we talked about free radicals and how dangerous they can be, and that they randomly give up electrons to other things. And so they have to be neutralized. However, because we're constantly creating them through regular processes like cellular respiration, we have to have an organelle that is going to neutralize them. That's what peroxisomes do. So peroxisomes neutralize free radicals by converting them to hydrogen peroxide. We all know what hydrogen peroxide is for the most part. Really good at destroying things, really good at sterilizing things. And so peroxisomes neutralize free radicals, converting them to hydrogen peroxide. This hydrogen peroxide is therefore can be dangerous when it accumulates. So catalases will convert hydrogen peroxide to water. So just to summarize one more time, peroxisomes convert free radicals and other harmful substances to hydrogen peroxide, which is then neutralized to water within the peroxisome. Peroxisomes can replicate by pinching off from pre-existing peroxisomes. In the liver and kidney cells, where we get a lot of detoxification, and we're constantly detoxifying harmful substances, peroxisomes are especially prevalent. Any questions? No. The next organelle is the cytoskeleton. It functions a lot like your skeleton does. It gives the cell shape, it gives it support, and allows for possible movement. And also, in the case of the cytoskeleton, it's also a road system or a traveling map. Within the cytoskeleton, there's 3 different types of rods. There's microtubules, which are made of tubulin molecules. hollow tubes made of tubulin molecules. There's microfilaments, which are made of actin subunits. And there are intermediate filaments. Microtubules, the ones that are made of tubulin, are the rods with the largest diameter. They radiate out from what are called the centrosomes, which are another organelle. They determine the overall shape of the cell and distribution of organelles. They also are key in directing chromosome movement during cell division, making sure that the chromosomes, the replicated chromosomes before division, ultimately end up in the new daughter cells equally divided. So little motor proteins carrying cellular structures use these microtubules as roads and pathways to move structures. These are magic roads in that they will assemble and disassemble constantly as they continually grow out of the centrosomes. So unlike our road system, they're constantly being assembled and disassembled. The microfilaments are the rods of the smallest diameter of the cytoskeleton. So you remember we had microtubules, microfilaments, and intermediate filaments. Microfilaments have the smallest diameter, and they're made of actin. They're involved in cell motility and changes in cell shape and strengthening the cell surface. These are found a lot at the cytoplasmic side of the plasma membrane. So right on the interior side of the plasmic membrane, where in that interior side of the plasmic membrane, they form what the terminal membrane or the membrane skeleton. These same microfilaments are also the actin filaments that are going to interact with myosin to form cleavage furrows during cytokinesis, so the last stage of cell division. They also are involved in interacting with myosin during muscle contraction. One more thing that they tend to do and can do is they will attach to cell adhesion molecules to allow for amoeboid motion through the formation of pseudopodia. These little, if you think pseudo meaning to look like, podia meaning foot, so these are false feet that are formed by the microfilaments that allow a cell to kind of adhere to a surface through the cell adhesion molecules, put out these little feelers and pull the rest of the cell forward, allowing a cell to migrate. Microfilaments are also involved in endocytosis and exocytosis. Remember how requires the deformation of the membrane, plasma membrane. Intermediate filaments are rods of intermediate diameter that are woven ropes with high tensile strength that attach to desmosomes and act as guy wires. Remember, desmosomes are the cell junctions that connect one cell to another. Act as desmosomes to resist pulling forces exerted on cells that can possibly separate cells. These are called neurofilaments in nerve cells and keratin filaments in epithelial cells. If you've talked about the skin in lab, or you're going to talk about it, these are the keratin filaments that keep cells attached to one another and keep them hard and have the protective function. Just a basic overview. Microfilaments are made of the subunits actin. They're the ones with the smallest diameter, 7 nanometers. Intermediate filaments are tough, insoluble protein fibers with a diameter of about 10 nanometers. These are the ones that are attached to desmosomes that help resist forces, tearing, shearing forces. And microtubules that are made of tubulin that are very important in cell division and transport of organelles throughout the cell. The centrosome, remember, this is what forms the microtubules. Are contained within them centrioles, which are made of 9 triplets of microtubules. So if you look here, there's 1 here, 2, 3, 4, 5, 6, 7, 8, 9. And each one of them is a triplet. So each centriole made of 9 triplets of microtubules.
So you know what? I'll just start parking on the wall.
And in many cells, in a resting cell, these centrioles are found within the centrosome at right angles to one another. Centrioles composed of a pinwheel of 9 triplets of microtubules. Centrioles function in generating the microtubules within the centrosome, organizing the mitotic spindle. If you think mitotic spindle, mitosis, this is the spindle that attaches to the chromosomes. And form the basal bodies of cilia and flagella. Cilia and flagella are the whip-like motile cellular extensions occurring in large numbers on the cellular surface. These are like little hairs on the surface of cells that allow substances to move in one direction across the cell surface.
I was curious too. Did your friends see it happen? Your neighbor?
Cilia will line the respiratory tract. If you think when you have a cold, you cough things up, then you swallow them when you're dealing with a cold.
You have to go back and find out.
The cilia help you to cough things up. When the central projections are significantly longer, they're called flagella. The sperm cell is the only human cell that contains a flagella.
Oh, all right.
Cilia will propel other substances across the cell surface, whereas the flagellum propels cells, the cell themselves. So cilia allow things to move across the cell surface. Flagellum allows the cell to move
And
itself.
that really caught my attention. Oh, my word, what was that? You know, and then it took me a minute to figure out what, so,
We have patterns of microtubules in the cilium and flagellum, that 9 pairs of doublets or 3 or 9 pairs of triplets that will encircle a center pair. Microtubule doublets are arms composed of dynein, a motor protein, that grips the doublets. Here we've got 2 doublets, grips the doublet and starts to crawl along its length, allowing one doublet to be bent. Resulting in the whole cilium bending. I'll show you something really quick.
thank you, Debbie, I really appreciate it. You'll get this. Bye-bye.
Cilia. Bending. Cilia surfing, let's put that in here. That sounds like a good one. We're looking at cilia here.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules.
Remember that the dyneins are the the dyneins are the motor proteins. So they were looking at the dyneins.
Demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism.
Now I'm gonna find one more. So the dynein were as they move down one, they're bending another one. Oh, let's see here.
The air we breathe contains foreign matter such as.
So cilia, like with the individual little dynein motor proteins, are going to cause the bending of the cilia. They're bending the microtubules within the cilia. And through this rhythmic bending, stroking pattern of the cilia, materials can be propelled along the surface of the cilia.
Dust, bacteria, and pollen. Hair-like cells called cilia, as well as goblet cells, can be found in the lining of the respiratory tract called.
So here's the cilia. Watch the way they have this stroking pattern of where things will be propelled along the surface. These foreign particles, these are bacteria, looks like it. And a mucus lining, and things are propelled along the surface.
Called the respiratory epithelium. Can be found in the lining of the respiratory tract called the respiratory epithelium. Goblet cells secrete viscous mucus that forms a layer.
Come back here.
Cells called cilia, as well as goblet cells, can be found in the lining of the respiratory tract called the respiratory epithelium.
So they do this little power stroke thing to propel the mucus along the surface. There's this alternating rhythmic beating between the power stroke and a recovery stroke that's going to create a pushing motion, allowing things to be moved along the surface in a traveling wave. So it bends down and then it flips up, allowing things to be moved along.
Dust, bacteria, and pollen. Hair-like cells called cilia, as well as goblet cells, can
Let me see.
be found in the lining of the respiratory tract called the respiratory epithelium.
This video, yeah, I like it because it's short, but I don't like the fact that you can't really
Hair-like
see the
cells
power
called cilia,
recovery stroke.
as well as goblet.
You can kind of see it here. So it's going to flip back and then it's going to flip up again. Why does this matter? If you've ever heard the smoker's cough, I think everyone's heard it at some point. smoking reduces the number of cilia. So there's fewer cilia to clean the lungs. As a result of the fact that there's fewer cilia, there's more mucus because your body has to figure out how to keep things clean. And if you have less cilia, your body's going to generate more mucus. Because the mucus isn't cleaned out as well, it tends to clog your airways and makes you cough. Because cilia also line the fallopian tubes and help sweep an egg cell toward the uterus, women who smoke have a higher risk of ectopic pregnancies, meaning fertilization that occurs outside of the fallopian tubes.
Is that also the same like as if you were to get pregnant and it gets stuck in the fallopian tube, or is that something totally different?
I think that's the same thing.
Okay,
I think from what I understand, an ectopic pregnancy is defined as any pregnancy where the embryo doesn't implant within the uterus. And so when the egg is not swept down the fallopian tube, it's going to implant somewhere else. Does that make sense?
yes. Now, would that be due to the hairs though? Like not being able
It
to?
could be due to the hairs. It could be due to different things.
Oh, okay.
But women who smoke are at higher risk because they have reduced cilia.
Okay.
Yeah, I mean, it's not just women who smoke who ever have ectopic pregnancies. There's always exceptions to things. So the primary function for microtubules inside a cell is what?
Is it A?
B.
It's actually B,
Okay.
because the maintaining the cell shape is sometimes a little bit microtubules, but a lot of it is the terminal web made by the microfilaments. And movement and contraction with the muscle cells is also the microfilaments made of the actin. The largest organelle of the cell is the nucleus. And you guys, actually, let's take a break. Let's take a 10-minute break. We will come back. We will finish chapter 3 and we will start chapter 7.
Sounds good. Thank you.
Okay. Hi, Mrs. What are you doing? Pretty cute. Mom,
Yeah.
come here. She just kind of loud.
It was witnessed.
What?
It was witnessed. the accident.
Oh, really?
Yeah.
Okay. Pupil teaching assistant. Is this really? Are you gonna take that out? Oh,
But I have that, here's my email and then all my text. We had quite a fun conversation. This Brian has so many issues now.
Just for a second, I will figure out.
Maybe we gotta get some breakfast. I know, I had it in my
You
hand.
just have it in your hand. Oh, come on.
There, see, I can't find it. I put it down in a strange place. I need other eyes.
Strange where it looks.
She said, I pulled up here and I thought, what? That's not where I am. I thought that's where I am. Then I realized, oh, back here. Yeah. To angle myself close to the sidewalk.
I don't think I ever really used those mirrors in all honesty. I always look over my shoulder.
You need to do both because of blind spots.
That's why I look completely over because I feel like my mirrors don't really show much.
Why?
there snooping around, I said, Casey, remember our responsibility? We still have class. And then she remembered.
And then she did both. No, but she was sleeping around. And I'm like, oh, we still have class. It's so busy.
I know, but we have
I have responsibilities out here. We have to check the perimeter.
we have
Other responsibilities. Oh. When you have time, I'll have to share my texting conversation with Brian. It's really quite interesting. He said he quit Christianity 40 years ago.
really.
And then he goes on from there, and everything he says is like, well, you're right on target there. It's like, and you're right on target there. There's one thing he said that I'm gonna have to gently correct over time, but I'm not touching it right now.
Okay, so the organelle, the last organelle, the main one is the nucleus, the gene containing control center. Contains the instructions needed to build almost all of the body's proteins. Cells can be uninucleate, meaning have one nuclei, be multinucleate, having multiple nuclei, or be anucleate, meaning they have no nucleus. Think A as being the absence of, so anucleate meaning that it does not have a nucleus. Cells like red blood cells that are anucleate means that they cannot create their own mRNA. They don't have their own DNA. So these cells cannot reproduce or be replenished. Three major recognizable structures of the nucleus are the nuclear envelope, the nucleoli or nucleolus, and chromatin. The nuclear envelope, remember that this is continuous with the membranes of the endoplasmic reticulum, has an outer nuclear membrane and an inner nuclear membrane. The inner nuclear membrane is lined by the nuclear lamina, a scaffolding of laminins of intermediate filament class. If you remember the intermediate filament class, actually let me back up just for a second here. If you remember the microfilaments that were made of actin that make up the terminal web of the cytoplasm, that is right next to the plasma membrane, the nuclear lamina is similar in its function in the nucleus. It maintains the shape of the nucleus and helps organize the DNA in the nucleus. Like the plasma membrane, this nuclear envelope is selectively permeable, allowing certain things to move through and blocking other things from moving through. but the passage of substances in the nucleus is much freer than it is in the plasma membrane. We have nuclear pores that create a channel regulating the movement of large particles in and out of the nucleus. mRNA molecules that are going to be synthesized into protein are transported through these nuclear pores. RNA molecule, let me say this differently. The ribosomal subunits, so if you remember, we talked about the ribosomes and how they're made of protein and rRNA, ribosomal RNA. So two things that are easily going to move through these nuclear pores are mRNA molecules and the ribosomal subunits. In the nucleoli is where the ribosomal subunits are created. So the nucleoli contain the DNA and the genetic material necessary to create the ribosomal RNA that will be used in ribosomal subunits. And both the ribosomal subunits and the mRNA are both going to exit the nucleus via the nuclear pores. In the nucleus, the chromatin and the nucleosomes are contained. The chromatin is a system of bumpy threads composed of 30% DNA, 60% globular histones, and 10% RNA chains. If you look here, here's the DNA double helix. It wraps around a core of 8 histones. So an octamer of 8 histones. And the DNA double helix wraps around this octamer of histones twice. When the DNA is wrapped around this octamer of histones, this structure is referred to as the nucleosome. So using an electron transmission microscope, the chromatin and the nucleosomes look like beads along a piece of thread, piece of string, with the nucleosomes being the beads. If you really wanted to study gene expression, which we touch on here, but we're not going to elaborate on it a lot. If you really wanted to study gene expression, you'd learn that the histones are vital to the regulation of which genes are expressed and which genes are silent. Based on whether the histones are methylated or acetylated. Do you remember the methyl functional group? Remember the methyl functional group? someone look back in chapter 2 and tell me what is the chemical property of the methyl group? So go back to the functional groups in chapter 2 and tell me what's the chemical properties of the methyl group.
It's a carbon atom surrounded by 3 hydrogen atoms.
Right? Is it polar or is it non-polar?
Non-polar.
Non-polar.
It's very non-polar. When the histones are methylated, meaning you add a methyl group to them, that gene is silent. So that means that that gene will not be expressed within that cell at that time. One of the main functions of the methyl group in general in biology is its role in DNA expression and gene expression. So it makes that DNA inaccessible. By adding that methyl group to the histones, you make that DNA inaccessible for gene expression. So condensed chromatin that's inaccessible for gene expression is inactive and it stains dark. When the DNA is acetylated instead of methylated, the acetyl group is a negatively charged negatively charged polar group. When the histones are acetylated instead of methylated, that DNA is open for gene expression. When we talk about cell differentiation, which is coming here in a few slides, this is key to whether or not a cell will become one type of cell versus another type of cell. It's all based on whether these histones are methylated or acetylated. If it's methylated, that gene is silent. It will not be expressed. If it's acetylated, it is expressed. If a gene is not expressed in a given cell, that cell may not become a particular type of cell and will become another type of cell instead. So that's why histones are crucial. There's a lot of ongoing research looking at histones because we know that they're the key to whether or not genes are expressed or not. When a cell is preparing to divide, the chromatin is going to condense into chromosomes. This prevents tanglement and breakage during cell division. Before the cell starts to prepare for mitosis, the DNA, the chromatin is loose string-like diffuse material within the cell. When it starts to compact up and become a chromosome is when you can actually see it as a chromosomal structure within a cell that's preparing to divide. So the chromatin has to compact up and condense into chromosomes before the cell can divide. So DNA, much of DNA is codes for different RNA molecules. There's 3 basic types of RNA. There's transfer RNA, which are involved in protein synthesis, binding one amino acid to the next amino acid via the catalyst of the ribosome. There's ribosomal RNA, which is part of the ribosomes themselves. And there's messenger RNA, which carries the DNA sequence that will ultimately be converted into a protein. So transcription is where the DNA is encoded into mRNA. And actually, let's not say just mRNA. Let me take that M out and say all RNA. The DNA within a given mRNA is then decoded and used to assemble a polypeptide in the cytoplasm. Polypeptide meaning, remember, a polypeptide is a protein. So a sequence of amino acids, AKA a protein. So DNA is converted to RNA via the DNA base pair rules. And I'm gonna stop sharing and use the whiteboard for a moment here. So DNA has the nitrogenous bases. DNA has the nitrogenous bases of adenine, thymine, guanine, and cytosine. Adenine base pairs with thymine. Thymine base pairs with adenine. Guanine base pairs with cytosine. And cytosine base pairs with guanine. Because they always base pair like this, and DNA is a double helix, anytime that adenine is on one sequence, thymine is on the complementary sequence. Anytime that thymine is found, adenine is on the complementary sequence at that same area, same spot. Guanine, same thing for cytosine. Cytosine, anytime you see a cytosine, guanine is on the complementary sequence at that same spot. When DNA is converted into RNA via transcription, thymine still base pairs with adenine. However, we don't have thymine as an RNA nucleotide, so we have uracil instead. And then guanine still base pairs with cytosine. And sorry, cytosine still base pairs with guanine, and guanine still base pairs with cytosine. So when we are taking the DNA and encoding it into RNA, thymine is going to base pair with adenine. Adenine is going to base pair with uracil. Cytosine is going to base pair with guanine, and guanine is going to base pair with cytosine. So a DNA sequence is going to dictate what the RNA sequence is. And the only major change is that we don't have thymine, we only have uracil. So based on these base pairing rules, we can always dictate what the DNA sequence will look like in the mRNA sequence. So the DNA template strand, taking this one right here, is going to transcribe into an mRNA strand that's A with U, C with G, C with G, A with U, A with U, A with U, C with G, C with G, G with C, A with U, G with C, T with A. So it's a 1-to-1 conversion over. The genetic code or the code that the mRNA is read into for proteins is a little different. It gets read as three base sequences called codons. Give me 1 second here. Go back to the whiteboard. So AUG codes for one amino acid. AAA codes for another amino acid. GCG codes for something else. UUU codes for something else, and so forth. So each set of three bases codes for one amino acid. Each of these three base pairs, three bases, are called a codon. So each codon codes for a given amino acid, for an amino acid and or a start codon where the protein sequence should start codon, or a stop codon. And each codon, just keep in mind, each codon is three bases. So coding DNA to RNA is a base-to-base pairing code. When we're converting the RNA into DNA, it's each three bases codes for one amino acid. Any questions about this? Oh, wait, let me make sure you can actually see it, because I know you can't see it right now. Any questions about this? Well, what if we have proteins that are misfolded? talked about the danger of accumulating misfolded proteins in chapter 2, at the very end. How do we get rid of them? Organelles can live out their usefulness and need to be degraded. They need to be, they're degraded by the lysosomes. Misfolded, damaged, or unneeded proteins are marked by what are called ubiquitin molecules. They're called ubiquitin because they're ubiquitous. They're everywhere. And these ubiquitin tags on a protein mark that protein for degradation by by proteasomes or proteases. Remember, proteases, ASE means it's an enzyme. Protease meaning that it's an enzyme that breaks down proteins. Proteasomes are a large enzyme structure that will break down proteins. So when misfolded, damaged, or unneeded proteins are in the cytosol, they are marked for destruction by adding a ubiquitin tag to it. And the proteasomes will then break down that protein. Some extracellular material. So now we talked about everything inside the cell. Let's take a second and talk about things that are outside of the cell. Body fluids function in transport and dissolving, such as interstitial fluid, blood plasma, and cerebrospinal fluid. Cellular secretions, such as intestinal and gastric juices, function in digestion. Saliva, mucus, and serous fluids function as lubricants. I have the extracellular matrix that's composed of proteins and polysaccharides. Remember, polysaccharides are polymers of carbohydrates that can be hard or soft and function as a cellular glue, a medium for cell signaling. I spent a lot of time with my first master's degree that I have doing cell culture. And one of the things, what I was doing at that time was trying to culture liver cells, mouse liver cells. And I was trying to use a protocol for culturing rat liver cells. And after probably about a year, it took me a year to figure this out, that there was no way I was going to use the same protocol to culture mouse liver cells using a rat liver cell protocol. I couldn't do it. And what would happen is that I would put my mouse cells, I would isolate them the same way that they did the rat cells. And then the cells I would put them on a dish and they would immediately de-differentiate. They would convert back to something that wasn't a mouse cell, and would take on the function of something completely different. And I can tell you it was really quite frustrating because they didn't die. They just no longer acted like mouse liver cells. And so it took me a while to realize that there was no way I was going to get these mouse liver cells to culture like rat liver cells. unless I changed something up and I figured something out. I figured out what was wrong, because what they do is they would just de-differentiate and not function as liver cells anymore. And I did some research, some period, what do you call it? Looked at a lot of research articles, looked at what other people were doing, how were they doing it, what could I be doing differently? And finally realized that I had to get an extracellular matrix involved. And so I had to plant these cells on an extracellular matrix to get them to function as liver cells. And when I did that, when I put them on this liver cell matrix, all of a sudden they started acting like liver cells again. And I don't know what it was about that matrix. I don't know if when you put them on the dish, the cells stopped communicating with each other. And therefore they would stop acting like liver cells, or if they needed that matrix to be in communication with each other to maintain their liver cell function. But I'll tell you that I struggled with this for about 2.5 years. And it was a very long 2.5 years, maybe actually closer to 1.5 years. But it took me a really long time to realize that I could not get them to function like, I mean, you would think mice and rats would be pretty similar. Last thing that we're going to talk about, I think it's one of the last things here in this chapter. Embryonic stem cells can differentiate into multiple different types of cells based on the chemical signals they receive. So as I was telling you about these cells de-differentiating, cells have to receive certain chemical signals to make them convert into specific types of cells and specialize in certain types of cells. Chemical signals can be slight in that you can have differences in oxygen and carbon dioxide concentrations. And cells can release chemicals that will influence the differentiation of neighboring tissues, cells, through altering the gene expression in neighboring cells. This has a lot to do with our cell adhesion molecules and receptors, integral proteins that determine what type of cell a particular cell is going to become. Cell differentiation is this development of specific and distinct characteristics in cells. Where we get increased amounts of particular organelles or actin-myosin. If you remember, we talked about how mitochondria will increase in its amounts in order to, are you guys seeing me at all? You're not seeing me, are you?
No.
Shoot.
No.
What happened?
Nope.
Really?
We can see the slide though,
But
just
you
not
you can't
you.
see me.
Right.
I was there initially, wasn't I?
Yes.
Yeah, you were.
Yeah.
Oh, wow. Do you still see me or hear me?
We can hear you.
We can hear you.
Just like, we're gonna try something. I have to remember how to do this. My dad tries to tell me. F9, is it F9? No. Er. Cancel. All right. Now I think I just made it worse. Let's see. I'm still not seeing. Mirror my video.
Can you try to do like Alt V?
Alt what?
On our screen, on my screen, it says if you start the video, it would be Alt V. Like
Alt V?
ALT, no, ALT, and then the letter V.
V is in Victor or T is in Tango or what?
V is in Victor.
Nope.
Nope. All right. Sorry.
Start video. What is that? Well, that's
Yeah, I was just
we
about to
just
say,
did.
hit start video. That should help you come back on.
Why isn't it not working? Alt V. Ah,
Do you see next to where it says mute at the bottom of your screen on the left-hand side where you can mute or unmute yourself?
Yep.
There's a start video.
Right.
you-
I'm trying to click that. It's not doing anything.
Is
Oh.
it letting you at least hit the arrow that's next? Like there should be an arrow in the box with start video, and you should be able to hit that and hit your camera setting, which is like, for instance, mine is the HP True Vision HD camera, and
All
it
right.
should
It
allow
says
you to hit that.
HD camera, original ratio HD.
Yeah.
Is there a check mark next to it?
They're both check marked.
Oh, that's awkward.
Well, I can get rid of them. I can take
Uncheck
them
it.
off.
I
I
know
just-
mine are checked, so I
Oh,
know
yours are
mine.
checked. Okay.
Yeah, so that's why I said it's pretty awkward, because usually if you're not on through Zoom, your video's not showing, you hit that arrow and you're to be able to hit the HP, like whatever your camera is.
All right. Well, if you can deal with me not being able to see me, I'm just gonna keep moving forward. Because, and I can figure this out afterwards. If you can deal with not being able to see me.
Yep, that works.
Okay. So embryonic stem cells, based on what chemical signals they receive, will differentiate into different cells. If you've heard a lot about stem cell therapy, stem cell, the morale behind it and so forth, this is what they're referring to. I have a friend who is in the process of getting stem cell therapy for lymphoma that has metastasized to his brain. And he's been struggling with it for probably about a year, maybe, and just recently reached the top of the list for undergoing stem cell therapy. And he's doing it probably as we speak. He's a professor at a school, I think in Illinois, either Illinois or Missouri. But we'll see what happens. Apoptosis. So this is cellular death, a process of controlled cellular suicide that eliminates stressed and unneeded aged or excessive cells. As damaged macromolecules or a chemical signal is going to activate a series of caspases that will destroy the DNA, the cytoskeleton, etc. The cells will shrink, detach from one another, round up, and start blebbing, meaning they just start bubbling up and breaking down. Dying cells will release chemicals. So these blebbing, shrinking, detaching, rounding up cells will release chemicals that will attract macrophages, and those cells will be phagocytized. The dead cells will be phagocytized. So sometimes we need to be able to get rid of cells when they're worn out, unneeded, or excessive numbers. Cancer cells can avoid apoptosis. They're acting strange, but they don't die because they avoid looking strange. However, heart muscle during heart attacks or nerve cells during strokes undergo apoptosis due to the oxygen deprivation signal. If you remember, one of the things that starts autolysis with lysosomes is an oxygen deprivation signal. So hypoxia, another word for oxygen deprivation is hypoxia, will start a process of apoptosis. Some changes involved with rates in cell division, or some words involved with the rates of cell division. Hyperplasia is accelerated growth. For example, in the case of anemia, bone marrow cells will undergo hyperplasia, so what red blood cells are produced at a faster rate. Atrophy is when there's a decrease in the size and number of the cells within an organ or body tissue. Muscles that lose their nerve supply often will atrophy and waste away. A lack of exercise can lead to thin and brittle bones or atrophy of those bones. The wear and tear theory is often attributed to cell aging due to chemical insults and the formation of free radicals. You remember we've talked a little bit about free radicals, how they're neutralized by peroxisomes. Well, they can still accumulate over time. And the result is that there's mistakes in DNA replication. Most free radicals are produced in mitochondria and energy production. And the result is that the production is diminished because of the radicals damaging the mitochondria. So free radicals that are produced in the mitochondria are going to cause destruction of the mitochondria. Also, temporary lacks of oxygen can cause, remember, this is hypoxia. So anytime that there's a reduced amount of oxygen, that's a hypoxic signal. Let me actually, because I've used this word twice, let me type it out for you. Hypoxia is reduced oxygen levels. Reduced oxygen. So if you remember, we talked about how the mitochondria are involved in aerobic respiration. So when there's reduced oxygen, there's reduced cellular respiration. Another thing that can be involved in the aging of cells is what's called the telomere clock. Telomeres are found on the ends of the chromosomes. They protect the chromosomes from getting frayed at the ends or fusing with other chromosomes. Each time the DNA is replicated, 50 to 100 of the end nucleotides are lost and the telomeres become shorter. So telomere clock will determine how many times a cell can divide. When the telomeres reach a certain minimum length, a stop division signal is given and those cells will no longer divide. So the telomere length is the clock and it's counting down. And once it gets to a certain length, the cell will no longer divide. If you think we have immortalized cell lines out there that will divide and divide and divide and divide and divide up until the end of time. Well, what they found is that in immortalized cell lines, their telomeres don't change in length. They don't get shorter. In cells that senesce and go into a state of where they will never divide again, the telomeres are at the minimum length. So that's the evidence behind this telomere clock. A disease called Hutchinson-Gilford progeria syndrome is caused by mutations in the gene that encodes for the nuclear matrix protein laminin A. Remember, this was one of the proteins along the internal surface of the nuclear envelope. Telomere length in fibroblasts, meaning the growth cells, are shorter than those in age-matched controls when looking at children with progeria and normal children. So their cells don't have the same capability of dividing and dividing and dividing. Their telomeres start at a shorter length. So their clock, they have less time on their telomere clock. And if you're familiar with this disease, this is when children age really fast. So the progeria hematopoietic cells don't express the laminin A with normal length telomeres, which limits how much their cells are able to divide, possibly related to how quickly these children age. Any questions? Okay. Any questions about chapter 3?
Yeah, it said our chapter 3 test was due last night.
It's not due last night. I posted it last night. It shouldn't be due last night. I posted it last night. It will be due in about 2 weeks.
Okay, I figured, but
it will open at about 1 p.m. today.
okay. I was just trying to play catch up without a computer, so I was like, oh my God, we didn't even
I
finish that.
understand. Okay. Other questions? So let's start now that we've spent time talking about chemicals, we've talked about atoms, we've talked about cells. Let's start actually looking at the real physiology. We needed that material. We needed this background material in order to start understanding real organs like bones and neurons and muscles. So that's what we're going to start right here. So bone tissue, chapter 7. The skeletal system consists of bones, cartilage, and ligaments tightly joined together. The cartilage is the forerunner of most bones in the embryo skeleton and bone development in childhood. Talk about how this is important at the growth plates in long bones, and that in at least one form of bone development, it is the precursor for it. It also covers the mature surfaces of joint surfaces. It covers the joint surfaces in mature skeletons, allowing for reduced friction at joints, or less or no friction at joints. We have ligaments that hold bones to bones at joints. We have tendons that hold muscles to bones. Both cartilage, ligaments, and tendons are all part of the skeletal system. Actually, let me back up this up. Tendons are not part. Cartilage and ligaments are. So because tendons are involved with muscles. So cartilage is a precursor for many bones, and it adheres to many bones. Ligaments hold bones to bones. Tendons hold muscles to bones. So tendons is actually not part of the skeletal system because it's involved with muscles. So bones, cartilage, and ligaments are part of the skeletal system. tendons that connect muscle to bone are not technically part of the skeletal system. So which of the following tissues could be found on or within a bone, such as the humerus? If you remember, we talked about connective tissue, we talked about nervous tissue, talked about cartilage, we talked about osseous connective tissue. All of these can be found in bone. So all nervous tissue, bone marrow, blood, connective tissue, bone, osseous tissue, some red bone marrow, some adipose tissue, all found in bone. Bones also continually remodel themselves and interact physiologically with other organ systems. They're your main storages for calcium and phosphate. And if you remember, we talked about how calcium was vital for muscle contraction. Therefore, each bone is considered an organ, being composed of different tissues that work together. Different functions of the skeleton. The skeleton is involved in supporting body, teeth, and viscera or organs, for example, intestines. It supports soft tissues, provides attachment points for tendons of the skeletal muscle. It protects and encloses internal organs such as the brain, the spinal cord, the heart, the lungs, the pelvic viscera, the bone marrow. It's involved in movements, including those involving the limbs, breathing, and other movements where muscles act on the bones. It's also key to balancing electrolytes through calcium and phosphate storage and their release into the tissue fluids. So inorganic phosphate, remember that functional group, the phosphate, its main storage is in the bone. We also, another function of the bone is that it's involved in acid-base balance. Remember, this is pH. Through helping buffer the blood against excessive pH changes by absorbing or releasing phosphate and carbonate salts. Blood formation also occurs within the red bone marrow, where blood and immune cells are produced. A hematopoietic tissue is a tissue in which new blood cells are formed. Let me stop just for a second here. Hemato or hema always means blood related. Poetic always means something being produced or something that's going to divide into something else. The progenitor cells that will develop into other things or divide into other things will develop or divide into other cells. Other cells. Okay. So hema always means blood. Poetic always means a progenitor cell, something progenitor that will develop or divide into other cells. So a hemopoietic cell is a cell that's going to divide into other blood cells. Which of the following functions of the skeletal system would be considered a metabolic function? Remember, metabolic has to do with chemical reactions. So metabolism is defined as the sum of all chemical reactions within the body.
I want to say support.
Why do you say that?
Because the skeletal system, I feel like, supports the structure of the body. So that's just a guess.
Okay. That's close. It's actually mineral storage, because if you think about how often minerals are involved in the protein structure, if you remember from chapter 2, minerals are very important in maintaining protein structure.
Mm-hmm.
And you have to maintain the protein structure to maintain the function. And if an enzyme doesn't have the right structure, or protein doesn't have the right structure because of trace minerals that are not present, the metabolic function will not occur. Different types of bones. We have long bones that have a greater length and width and consist of a shaft and variable numbers of ends. We have short bones, like the carpal bones, that are somewhat cube-shaped and nearly equal in length and width. We have flat bones that are generally thin and afford considerable protection, like the bones of the cranium. We have irregular bones, like our vertebrae, that have complex shapes and can't be grouped into any of the other categories. In short, flat, and irregular bones, thin plates of periosteum, which is dense, irregular connective tissue, covers the compact or cortical hard part of the bone on both sides. Endosteum covers the interior bone. So let me add some more terms really quick here. I'm going to clear all our drawings. Peri always means around or on the periphery or on the periphery or surrounding. Enda always mean, if you remember, we saw this when we talked about an endergonic reaction, talked about endocytosis. Endo always means inside. Ostium, anytime that you hear the word ostium, it means bone. So periosteum is a dense, irregular, or where the periosteum dense, irregular connective tissue that lines the outside of bones. Endosteum is dense, irregular connective tissue that lines the inside of bones. With that in mind, we have compact bone on the outside. We have spongy or cancellous bone on the inside. Periosteum lines the compact bone. Endosteum lines the inside bone. Where an inside in the cancellous spongy bone, it looks kind of spongy. That's why it's called spongy bone. In between what are called trabeculae, trabeculae are little bars, trabeculae, there is bone marrow. The red bone marrow is located in the trabecular cavities, so these little inner spaces of the spongy bone. So cortical or compact bone is the dense white osseous tissue. That outlines the bones. It encloses the marrow cavity or the medullary cavity. We're going to learn a lot about medullary in anatomy and physiology. Medullary means inside. You're going to see this word fairly often. Medulla is medullary. So medullary means a cavity, internal cavity, or internal area. Let's just say internal area. So cortical bone line is the outer shell. Spongy or cancellous bone is in the central space of long bones and at the ends. The diaphysis of a bone is the shaft of the bone, of a long bone. So it's this whole long region right here. The epiphysis, we have two epiphyses on each side of the bone. Each end of the bone is called the epiphysis. The diaphysis meaning in between. So epiphyses are the distal and proximal ends of the bone. The metaphysis is where the diaphysis, this long shaft, the metaphysis is where the diaphysis meets with the epiphysis. You gotta love all these physis words.
Yeah.
So metaphysis is where the diaphysis meets up with the epiphysis. The epiphyseal or growth plate is the hyaline cartilage. If you remember from lab, this is one of your connective tissues. Have you gone over connective tissues and cartilage in lab?
Yeah.
Say yeah, yes, Professor Gessel, we have.
Yes.
Okay. The hyaline cartilage in the metaphysis is where growth of the long bones is going to occur. Or we're going to get elongation of long bones. When growth of the long bone stops, an epiphyseal line will form here and seal these plates. So here's my epiphyseal line where my epiphyseal plate has formed. Another place where I have hyaline cartilage, so I have it in the epiphyseal plate. I also have it on the surfaces of the epiphyses, epiphyses. Because it is, if you remember, one of the reason why we call hyaline cartilage is it's glassy. It's very smooth in its surface. It allows for a very smooth articulating surface for joints. So we have an internal medullary cavity within the diaphysis that contains, in adults, contains yellow bone marrow, which is a fatty tissue that can act as a reserve fuel supply. The endosteum is a layer of reticular connective tissue that's lining this inner medullary cavity. In the endosteum, there are cells that dissolve the osseous tissue and cells that deposit it. We'll talk about what those cells are called next Friday. Also, within the outer surfaces of the bone, there are nutrient foramina. Foramina means holes. Stay with me for 5 more minutes here. Foramen means hole. Foramina is the plural form of holes. When you learn about bony structures in lab, remember that foramen means holes, foramina means holes. So foramen means hole, foramina is the plural form of holes. So we have teeny holes in the periosteum and the cortical bone where blood vessels enter the bone. The periosteum is this sheath of tough, fibrous, dense, irregular connective tissue that is the inner osteogenic layer. If you remember, I think we've talked about this. Genesis means growth of new things, creation of new things. Genic means growth of new things. So osteogenic meaning growth of new bone. So the periosteum is this fibrous collagenous layer with an inner osteogenic layer of new bone forming cells. Some collagen fibers are continuous with tendons. Remember, tendons can connect muscles to bones. Some collagen fibers in the periosteum are perforating fibers called Sharpie's fibers that are going to insert into the bony matrix of the cortical bone. So a general feature of flat bones is that they have this. So before I keep going, any questions about the general anatomy of long bones? General features of flat bones is that they're sandwich-like. They have a sandwich-like construction where we have an outer surface of compact bone. We have an inner surface of compact bone. And in between, we have this spongy bone, or this called diploie. Of spongy bone. The diploie functions to absorb shock and has marrow spaces lined with endosteum. I think this is a really good place to stop. Do you have any questions for me right now?
Nope.
Okay. Have a wonderful weekend.
You as well. Thank you.
Beautiful. Take care.
I hope you start getting excited because now we're actually going to use all this stuff that we've talked about, all this monotony that we've talked about with regards to atoms and ions and cells and blah, 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 blah. Now we're actually going to say, okay, how does this actually relate to the human body? So have a wonderful weekend.
Thank
You
you.
too. Thank
You
you.
too.
Bye-bye.
Ariana or Lydia, are you there?
Yes, but I'm heading out.
Hi.
Thank you.
Okay. Ariana, are you there?